Hello, my name is Skylar, and today we will be studying trait linkage in strawberry poison dart frogs. Here on the left, we have the heterozygous F1 generation frog with the phenotypes long tongue, spots, and blue legs, produced from the pure breed cross between two homozygous frogs, the top having a short tongue, spots, and no blue legs, and the bottom having a long tongue, no spots, and blue legs. Here's the map of the heterozygous F1 frog. The distance between the first two loci, or R1, is 15 centimorgans, while the distance between the second and third loci, or R2, is 10 centimorgans. The coefficient of coincidence value is 0.84. To show how the 8 possible gamete types are produced from the F1 progeny, I will be using pipe cleaners and beads as a demonstration. Here is what no crossover event would look like between two parental chromosomes, resulting in gametes with either the genotype little a big B little d or big A little b big D, with the probability being 0.3818 for either of these combinations. Next, we have a double crossover event with crossing over occurring at both R1 and R2. You can see how the pipe cleaners are wrapped around each other at these locations to portray this, resulting in the formation of two recombinant types, either little a, little b, little d, or big A, big B, big D, with a probability of 0.0063 of either of these occurring. The formula for the probability of the double crossover is R1 times R2 times C, the coefficient of coincidence, and once you plug your numbers in, you get 0.0126. That times 1/2 is 0.0063, which is the probability for each gamete. Here's what a single crossover event at R1 would look like, resulting in two recombinant types, either big A, little b, little d, or little a, little b, big D, with a probability of 0.0687 for each of these. We obtain this by using the formula for the probability of a single crossover event at R1, which is R1 minus the value for the probability of a double crossover event, which we calculated previously. Once we plug our numbers in, we get 0.1374. That times 1/2 is the probability for either gamete. Here is another example of a single crossover event, except this time it's at the R2 position, resulting in two recombinant types, little a, big B, big D, or big A, little b, little d, with a probability of 0.0437 of either of these occurring. The reason why this probability is smaller than the probability of a single crossover event at R1 is because the map distance at R2 is shorter than at R1.